Good morning, everybody. If you don't know me, my name is Dwayne Sador. I run the social media for Carlisle Liquid Starters. Today with us is Scott Walker. Scott needs no introduction. If you don't know him, he's all about the data. And today, Scott is going to talk about us with biologicals. Take it away, Scotty.
Good morning, folks. Thanks very much, ladies and gentlemen, for the opportunity to bring this webcast to you on biologicals and biological stimulants. As you can see, I have a company called DSW. It just stands for Donald Scott Walker, nothing fancy. And right off the bat, I'd like to take a moment and introduce and thank our host, Carlisle Liquid Starters. Daryl and I have been joined at the hip, so to speak, for some 18 years since moving out here many years ago to introduce a Nortrace foliar product line for UAP. He gave me my first start to be independent to develop this company called DSW and all the research that's unfolded, and hence the focus that's taken on this whole process of research and development of biologicals, biostimulants. But the key here is, how do we recognize them as we move forward, and how do we generate the greatest ROI from their use? Now, going forward, that seems like a fairly simple statement, but it takes time. And to move forward and develop them, it's going to take a lot more research to come up yet. So let's move into what the Europeans started some years ago that brought the interest in me traveling around the world to go to the various biological stimulants. So you see a definition as what it means to be a biostimulant. And it's very straightforward, but they did this in an effort to structure the development of labels. As you may understand, in our country, we've got the fertilizer supplement issue going on, and it's been very difficult to be able to put things on labels. Now, the European Union is so far along with this that by the year of 2022, they will have established a multitude of required protocols that different products, whether they're a biological and or a stimulant, will be required to go through scientific collection of data. Some of those research papers are anywhere between $50,000 and $75,000. So in an effort to be able to create some kind of an environment in Canada to offer that same kind of coordination of label structures, there's a brand new organization referred to now today called the Fertilizers and Supplements Advisory Committee. You'll see a list of items there that committee has been developed to do. And we've already started some of the working groups that were established on January 17th, this past January 2020. And we're working alongside the CFIA, as identified clearly here in what we do. But on these working groups, we also have members of the Pesticide Management Regulatory Association there to be a sounding board so that we don't try and reinvent the wheel. I know from the first few meetings we've had, I'm on the board of directors here, that we can see it's going to take some time to be able to get the regulatory bodies in line with what we're going to do going forward. The reason I got involved is for instance, I was one of 10, maybe 10 Canadians at the last World Congress that was held in Barcelona, Spain. And that event hosted 1,600 world delegates, head scientists from all parts of the world, and CEOs from various companies were there in attendance. And it was rather mind-boggling to be a part of that, but it really brings a focus on what it means that as Canadians, what we can do going forward to create these different things. So due to a limited time here, I'm going to jump right in. One of the products that I'm researching right now, I'm on contract with them, is the Advanced Ag has hired me on to help them with their R&D work of a brand new biological that stems together 5 different types of bacteria. So this would be along the lines of a biological as opposed to a biostimulant. And they're up and coming. They're 20 years in the making of their water products, 5 years in the beginning stages of an agriculture product. The key phrase to identify here at the bottom is found a way to stabilize, mix, and train up to 6 different effective strains of bacteria in one product. And I know in my travels and on my research, this is a rare event. Most often you might see 2, sometimes 3 if they include rhizobia, and different biologicals together. They're very difficult to stabilize, not to mention this chap has found bacteria. Oh, I've been muted by the host.
Scott, you're good to go.
Okay, there we go. So once again, I'll move right along here due to time. Just to identify the fact that these guys are very scientific. This is one of the research papers that was done through the International Journal of Research and it was published. I spent several days with them about a month and a half ago before travel restrictions occurred. And there I got together with a bunch of faculty members of the Lethbridge University, along with their scientists and a group of others, to try and nail down how do we move forward to create more scientific background to products of this nature, this one in particular. Because as you noticed in the prior screen, he's got up to 6 different types that he can stabilize together. He's moving forward and identified 2 more yet. And some of that research we're doing is to identify some of the functions that they've been able to generate in the soil. One of the reasons that he puts in there to be able to stabilize them and put them together, when you look at this type of chart, you've got bacteria A within 5 hours, it has been able to multiply itself to the tune of 1,024. Whereas a type of bacteria B, for instance, as an example, it's itself to that point of 32 different types of 32 counts.
We don't see your screen. Can you share your screen?
I certainly can. That's Alt-S.
Yes.
Okay, you got it? Can you see my screen now?
Nope.
Control.
No, Alt-S.
Alt-S. Can you see it now?
Nope.
Not sure what happened there, Captain. Can you see it now?
I can see it, Scotty.
You can see it?
Yeah.
Okay, sorry about that. Let's continue on.
Okay, back to this screen. You can see why it's such a difficult endeavor when you put different types of biologicals together, whether they're bacteria, whether they're a fungi, whether an amoeba, which is a type of food. It's very difficult to figure out how to balance and grow them together. And these gentlemen, to the Advanced Ag as well as Blue Planet and John Wong, they found a way to stabilize these 5 bacteria in a single environment. Now on the bottom left, you'll see a petri dish there and a halo around some kind of residue that's in the very center. That's one of the things that identify which particular types of material actually solubilize phosphorus the fastest. I'm going to take an example of the Bacillus subtilis at the bottom of that list. Now, you can take one Bacillus subtilis that's not been manipulated against another of the exact same strain, and they do not perform exactly the same. So in his efforts to make sure that these not only live and breathe and work together, but they actually have certain functions. And you'll see that there's different roles each one of these play. Now, as a producer in the industry, as a farm producer, you're really after their functionality, whether it's bacteria or fungi. It's the environment they create when they're applied in that growing zone that we're trying to utilize their benefits. And as we move forward, you'll start to see that there's a number of crops around the world. These are not just crops that have been tested here in North America, but they've been tested in all types of regions around the world.
Scotty?
Yes.
We're getting reports that nobody's seeing your actual screen though. They're seeing you. So
Fine.
Can you go to take your mouse and go down and you should see the share screen button.
No, not when I've got the, okay, I'll have to escape out of this. Okay. And I'll have to do this. Okay, go down here, which left or right?
It should be, if you take your mouse, it should be right in the middle. There should be a green button that says
Yep.
share
Yep.
Screen. Click that,
Go.
and then I'll show you a couple different screens. What do you want to share?
Okay, would be this one. Then hit share.
And then there we go.
I apologize, folks. Okay,
And then just make that full screen.
you betcha. We're back up and running.
We are perfect. Give her.
Okay, I'll go back to this screen briefly for those that were curious what I was saying about multiplying bacteria when they're together. And that's why there's a process, like Daryl Carlisle's a qualified brewer, where he's got a brewing management facility there where he actually has it all utilized within the realms of his computer system. so that he can track and develop these 5 bacteria together. For those that couldn't see the screen before, there it is there. These are very prominent bacteria that are in our soils, but through our cultural practices over time, have not got the populations that they could, not to mention, once I go back again to the functionality that John Wong has created in these materials. Anybody in the world can put these together, but they can't make them respond and work the way that John Wong has manipulated them to do so. Again, I'll go back, I'll jump ahead now to these crops that has been tested on. Moving forward, we're working with cereal crops and like in Western Canada. What I'm seeing from the research we're doing here in Canada, and that's a research farm there in southern Alberta where I'm doing replicated plots, is we're testing it on a number of crops, which takes time. Right now, these replicated plots that you see are involved in barley and then rotated into a crop of peas and then into a crop of canola. So we have 3 specific crops for Western Canada where they're testing them. That same square that you can see in that photograph is the exact same square that you used the following year, like in 2019, and it was peas. And in 2020, the exact same square will have the exact same treatment now, 3 years in a row, and this year it'll move into canola. Now, I'll give you an idea of what we're discovering as we move forward in all this stringent testing, is it doesn't work the same on all crops. We're all looking for silver bullets to say I can use one product that fits all, but in the world of biologicals, that is not the case, because even amongst the same species and different varieties, for instance, you look at that barley there, there'll be a different grouping of varieties within that class of cereal. They will not all respond exactly the same because the exudates that are generated by the plant while it grows are differentiated. And that's what feeds the biologicals in the soil. So when you look at these different bacteria, they will be supported by different crops. And what I've discovered to date, in particular, is when I'm doing the work with the canola, for instance, I'm not receiving the return on investment required to promote it deeply and excessively on canola. But on the other hand, pulse crops, legumes, and cereals are responding extremely well. The key is to identify a multitude of years. So first year in, we see the response in these plots in barley with different application methods is very significant. And yet in 2019, these numbers softened a great deal. And yet peas in the environment worked really well. So what we've got to do is move forward to try to identify the consistency of these types of responses with the utilization. Now, that being said, again, you look at the bottom there, one of those treatments on the peas was a 3rd node, and then the other one beside it was in furrow and then third node. At this stage, in discussions with likes of Daryl Carlisle and others that are helping me coordinate field research, is we're seeing that in furrow is the most consistent response. What we've got to do now moving forward is continue to do trial work, not en masse, but in small groups, to identify the consistency that we can get. Because as we move down the line here, we'll see one farm was able to produce almost 10 bushels. And then here, I'm working with it to identify its ability to grow through saline pockets. On the right of this flag is not treated. And that moves down through a saline patch of that field. Where on the left-hand side, it was treated. It was applied at planting in furrow with this G22, and you can see a substantial significance in the growth of that crop. And it was like that from beginning to end. And what we identified here when we compared the treated to a non-treated area of no saline is we managed to bring the yield up in that saline area to equal the field average. Not gigantic. I don't think we saw more than maybe 6-7 bushels in that effort, but significant that we're moving closer and closer to identifying ways and means to overcome some of these saline pocket issues. Then you move to another grower study where we did peas. You'll see on the left-hand side that we identified the fact there was a heavily manured section of peas that was part of this trial. Unfortunately, that area that was heavily manured for 2 years, some years ago, all went down before it got a chance to finish. So when we did weights and measures in that area, there was no significant gain on that return on investment. Whereas in the other section at the other end, it was a 2-section field, 2 miles down the road, where we were keeping an eye on the weights and measures. we're gaining anywhere between 12 and 10 and 15 bushels from its application because it didn't go down and it finished properly. Again, I want to make sure that I let you know that we're still in the working phases of developing consistent responses. Now here we see barley and also some wheat. And when you look at the number of heads, and this is the amazing part of research and trial work, and why I'm always pushing the aspect of limiting the length of those weights and measures. My weights and measures run anywhere between 500 and 800 feet. Now, my effort there is to quantify what the yield gain is. You look at the picture in the middle, the treated looks like it's going to be more than double of the non-treated. When in actual fact, the weights and measures of that wheat field showed that it was a mere 15% to 16% increase, not double as the picture would indicate. There are many biologicals and biostimulants that will make a crop look picture perfect, gorgeous in all our efforts, but it doesn't always come out at the other end in terms of weights and measures. But still significant to have that opportunity. But a keynote here, and I had a discussion yesterday in Daryl's office about this type of work, is in the lower areas of the field where the soil is much richer, the increase was not nearly as significant as it was on the upper slopes or the eroded knolls, in which case it was well above and into that 15% gain. The other interesting thing that I noticed on several occasions was working with the soybeans in the field, because I don't have that replicated in research yet. We're trying to identify what crops are worth pursuing and spending more money on. And the beans and the phaseolus beans or the legume beans, and soybeans in particular, we didn't see any serious growth differentiation until it got to the point where it went into the R1 stage, started to do the reproduction, it began to branch, it created more flowers, excuse me. And then when we went in to do the weights and measures, we can see here that when we first counted bean pods and everything, again, it looked like, oh my goodness, we're gonna double the yield, when in actual fact, you can see here it was a moderate gain of around 7 bushels. And again, that's really important to note when we're doing these field trial studies and you're introducing it into your operation, is where is the best fit and how do we get to achieve that? Now here you see an alfalfa study up in the Gladstone area, thanks to Mike Besser and his brother and father's efforts. This was applied, and we can see here again a very significant increase. And with these thoughts in mind, wherever I was involved in a hay trial, whether it got weighed or not, the responses in turf and pasture and hay production is nothing short of wow factors. I've never seen the likes of any biological I'm working with that can have this opportunity. These are significant. So this is another thing. Golf, as many of you know, is one of my diehard sports. And this is a turf application. You'll see on the right, this is prior to application, post a PGA golf tournament where the pros had beaten the living daylights out of the tee block. It was applied at significant rates, 5 to 7 gallons per acre, straight product, no water. Then then you see the photograph on the right. That was 10 days later in the fall, in October, temperatures ranging anywhere between 15 and 18 degrees Celsius. So not rapid summer growth, no fertility added, simply the biologicals moving into that soil environment where they're able to solubilize different forms of potash, phos, and accumulate nitrogen. Some out of the air, some breaking it down from the organic matter in that environment. So when you look at this, do we require a boatload of fertility when we do these applications? At this stage, early on in the research and study, it would indicate fertility requirements not needed to achieve these types of responses. Now that being said, I don't want to bleed the soil dry of nutrition, because alfalfa in particular can pull out a gigantic amount of potash, just phenomenal levels. So how long can you sustain that without continuing to feed those environments? That remains to be seen. The last thing on this particular product I want to draw to your attention, and this is something I've never seen before, and this goes along line with those that are using things like the G22 starter, where everyone says we're bleeding our phosphorus from our soil because we're not applying enough to sustain the levels that we're removing. Granted, you can't take $5 out of the bank today and go back and get it tomorrow. It's gone, it's gone. But on the other hand, what we see across Western Canada and certain parts of the Great Northern Plains of the US, is that our phosphorus solubility in our soil is inherently low. It's not identifying the fact that we have low phosphorus mineral in that soil. It's simply what the biological environment has been able to solubilize, stabilize an accessibility to the crop and moving forward. I had this soil sample done 3 times because the first time I would not believe it. So what this pasture is a bull pasture with a fence down the middle. The bulls are moved from one side to the other throughout the season to graze on the pasture. Only one side of the line, which is about 2-acre patch, was treated for 3 years, twice a year at that 1.75 gallons. There's never been any fertility put on that. They're only about 60 feet apart, these samples, on either side of the fence. And that's the differentiation that's created. Can we do that in our field crops that only grow for 3 or 4 months of the season? I think not. This is a homogeneous environment, never disturbed for some 20 years. It's got a reasonable pH, almost a little on the low side, right? Where bacteria generally don't survive strongly. So I've been able to kick up the solubility on a regular basis by applying it over and over again. But this pasture grows 10 months a year and goes into a dormant period that's very short. How long will it take us to achieve this in a field crop? Totally unknown. Will we ever do it? I don't know that. But it certainly identifies the fact that these 5 bacteria have the ability to sustain and create the solubility of a lot of mineralization in our soils to create the availability. Will we need to continue to address the removal? Absolutely. But in the interim, the whole idea is to achieve ROI on the application as well as have long-term significant benefits. Because some of the work I'm doing with these biologicals in this particular company is I'm now pursuing research on nematode control for potato production, on Fusarium head blight control in cereal production. And now I've seen and come across a series of emails that's starting to identify insect controls as well as suppression. This is the beginning of the very beginning edge of many of these to come. They are not foo-foo dust. You don't spend $50,000 to $75,000 per project on something like this if it's not got merit. And that's why you hear me refer to a Scotty data walker. It's not simply the research that's going into it for ROI, which as a producer is the only reason you would ever pursue these things, but it's also the long-term effect. Because we also got to keep in mind as I break into the next section, what a biostimulant can do. So this next product here has been around and I've researched it for a significant number of years. This product is simply a product that has the ability to generate a significant level of mineralization from the research that's done at universities, as well as photosynthetic response by its application as a foliar. So in an effort to move through quickly as to what are the main key elements of a biostimulant that such as this can do for us. Well, it's been scientifically quantified that after a foliar application, the synthetic response to photosynthesis is maxed out, increased significantly. When used with fertilizers, for instance, like UAN, the mineralization is significant. But these are substantiated through university trials, not hearsay. Standability is something I identified with all my field work and research work that I saw it over and over again. You'll see in some slides going forward why it does that. The other part for the producer is quality and yield. That's what it's all about for us in the field. So what is Lignagil? It's a manufactured biopolymer energy source, which is derived from the pulp and paper industry. In an effort to make paper, you have to remove the lignans, which are black, and this is simply a byproduct that comes from the pulp and paper industry. Now, when it first comes from that industry, it's like a hockey puck in water, has no solubility. You can hardly move it into containers to get it out of the tailing ponds. These gentlemen, these scientists found a way to react it with oxygen. They put it through a 6-stage process that's all been patented, never been reverse engineered. And all it simply is energy that has been extracted from the plant-based lignans. We consume solar energy in the process of photosynthesis that's laid down as lignans in the tree, and it's simply measured in the form of kilojoules of energy. There's a multitude of products with a multitude of application opportunities. You can see there in the soil as a foliar, as in seed. So basically, the key factor in foliar is that 32% organic carbons of these lignans aid the plants in its ability to increase photosynthesis, simply because we're adding more energy to that crop at that stage in that tissue. This has been supported to a multitude of years of research. I came across a paper when I was looking it up. That was 2012 is when they started that photosynthesis. So here, for instance, is a quick slide on the processes involved through photosynthesis, where the sun triggers the process in the leaf through its solar energy. But this is the nuts and bolts of what's created. You can see a significant increase, be it even branches or pods or stems or the leaves, it's significant. Now, this has several implications. Some are the exudates are then flowing through the root system, feeding the biological response, hence it's a stimulant, as well as any other enhanced efforts it can create in the tissue itself. So what does that mean? This is a perfect example. This was done all it simply does is generate a higher level of photosynthetic production. Those photosynthetic production elements, it then drive through the root system, as I mentioned, through the exudates, feeds the soil biology at the request of the plant. The soil biology responds then by releasing the food, and the plant goes on a feeding frenzy. And that's where the yield enhancement goes. Did I get a boatload of yield from this application? No, it was not timed at a reproductive state that would generate a boatload of yield. That's where the timings come in that have been structured over, I've got over 12 years of research, upwards of 14 now since I was first introduced to the product. It takes a long time. But the key element for us going forward as we look for these silver bullets is to identify where are we thinking? Are we looking at the top of the crop because that's where we get paid? Or are we looking at what substantiates that growth that creates that yield? And that is in the soil environment. I can't generate that without this. And when I can put different things together and identify the benefits, it all starts to unfold into yield. Excuse me. So moving forward, here's another byproduct of these exudates. As you can see here in corn, maize, it increases the level of fungi underneath that crop upwards of 58%, whereas the bacteria enhancement is 39%. Then when you look at the bottom below that, you'll see again, soybeans are a little bit different creature, but look at the amount of fungal response and bacteria response. They're a bit different, but that's primarily from the exudates that go into the soil from the plant at the bequest of the plant into that microbiology in the soil that then in turns is supported to create that environment below that generates this whole process and this whole paradigm shift of thinking of down below. If I can mitigate the level of disease pressure that's in the soil, so I have less sporulation, I'll have less sclerotinia, I'll have less fusarium head blight, all these things are attributed to the whole process. And that's my goal going forward, to see what else can we do. This is just a paper that was done some years ago over in the Czech Republic here, Prague, you'll see, to identify why it responds the way it does when it's added to fertilizer. So you can quantify it scientifically. Why do you see the increase in yield when it's added to things like, for instance, 28, because here it's a liquid in Canada. So here's, what does it mean as a producer? In this photograph, and I've used this photograph since 2012, and this occurs off and on, you'll see on the right-hand side, the crop is darker, lusher, greener, and that's what a producer loves to see. When actual fact, the lignajule is with the 28 side banded on the left. And you go, oh my goodness, it's lagging, but it's not. When I took a tissue sample of that particular zone, there were equal levels of nitrogen. But on the left-hand side, there was a significant reduction in potash, copper, and boron. And because of the lack of potash, copper, and boron on the right, the pigment development is different. And that's what happens when you over-apply nitrogen and it goes into a luxury feeding. You end up with a succulent plant susceptible to disease, A succulent plant that's susceptible to drought, as well as susceptible to lodging because of the lack of the potash. Moving forward, this is an area of saline low pH soil, a greenhouse treatment trial I was working on. This was also involved in some calcium work. The treatment was on the right, and you can see a significant difference in the standability simply because its ability to mineralize product and stabilize the salts to allow that crop to grow normally. So what does it mean when you see the pigment or the photosynthetic response on a stem? And this is where it really turns cool. As you look at the different slides there, the upper portions of the barley or wheat are untreated, the lower ones are treated. And that's a double application at what we would consider in that range of 40 grams an acre, which is a half a liter. And that stem is green, therefore it's creating photosynthesis. And if I can do that, you'll see the outer layer of that stem, I lay down more significant development of lignans. And that's why I get the standability. Not to mention the vascular bundles that are moving all the different elements of proteins and enzymes and growth regulators and plant food throughout that system. The vascular bundles are far more definitive and do a far more efficient job of the whole process. Here is what it looks like up close. It seems to be a bit more significant on barley than other cereals, but that's simply because barley of all the cereals is the worst at, I guess you would say, generating photosynthesis. It seems to be a bit of a dog. It doesn't do as near efficient job of generating photosynthesis as wheat or oats. Hence, it does a more significant job of helping your barley crops. Here's an example in the field where it was put in an airplane. You'll see a wiggly line going down through the field above the arrow. That's one application at heading with a fungicide, generating a stronger stem so that the heads don't bend over and the crop doesn't lean and lodge near his butt. What does that mean? If it's timed right with the right conditions, with good soil moisture conditions, there can be yield and almost always standability is generated. It's not going to hold the crop up in 6 inches of snow, but under a normal crop load, if you apply it twice, your standability is significantly increased. So what are some of the data points before I lose and run out of time? Here's some work that started back in 16. I've been working with Mara. These are third-party replications of various applications, whether it's with 28 all the way through. Its ability to generate ROI when it's added to nitrogen is significant. I'd love to put it on urea, but it's a humectant and I have to use a dryer, and logistically it's very difficult to get done and get to the field and get applied. So by and large, it's always been used with 28. And they're significant. Those are also field tested and done as well. Here's a slide there shows what it's like when it's put with canola with a herbicide. Is it the first herbicide? Is it the second herbicide? That swings and changes according to environmental conditions. And I've been trying it on either or, and those data points swing back and forth, back and forth. So I still have to admit though, I'd be more inclined to use it with my later herbicide on canola. That seems to be where I generally get the most significant increase. Now, this is an overall view of all the different types of applications. And these, remember, are all from plot structures at third-party research. These aren't my numbers. These are ones that I've hired out various facilities across the Western Canada to perform the trials. So these kinds of slides and thoughts in mind, what I look for in terms of statistical analysis with short weights and measures, be it in the field or if it's in the replicated trials, is 70% of the time I'm looking for an 8 to 12% yield bump. And is that from 1 or 2 applications? It can vary. And then I look for 15% of the time, I get what I call wild cards. For instance, the second bar graph in at 15.76 bushels, I call that a wild card. That is gigantic. That is rare. It happens both in the field as well as in the research, but it's only 15% of the time. The other 15% of the time, it's a dog. I can barely get enough yield that it pays for product. So it's like anything else in the world. It has substantial opportunities, but it becomes a management tool. It's not a silver bullet. It's something that you can look forward to. I'm going to be bringing into my fold again this year, testing 2 more biostimulants, one from Europe. I'm trying to get one from Australia, and it's going to take time, but I've probably tested 10 to 12 various types of biostimulants and biologicals but these two in particular seem to offer the greatest opportunity. And I'll leave you with this paradigm shift before we go to the questions and answers. Keep in mind, our crops are produced by the efficiencies of what we develop in our soil. Everything we can do to enhance the ability of that soil to do a better job will push back against diseases, will push back against insects, and when through that, those efficiencies will generate a higher yield and quality. It's not as though there are spaceships flying over our crops, sprinkling spores of disease. All those disease mechanisms are coming from our cropping practices that we've generated to create that host environment, to allow them to sporulate and pollute our crops. And then we move into the world of using fungicides. As soon as I go out there with a half-rate fungicide or herbicide time, how much damage do I do to my beneficial bacteria on the surface of that soil? I have no idea. Nobody's willing to spend the money. I have my suspicions. So anything we can do to mitigate the destruction of the beneficial populations in our soil, the more rampant and more push forward we'll be able to get down the road of direct regenerative farming. I am now open for any questions.
Does anybody have any questions for Scotty? Everybody's unmuted now. If anybody has any questions, now's the time.
If we're gonna have X, we're gonna get our timing on wheat down. I'm gonna ask Jeremy about that.
Can you speak up, please? I could barely hear you.
You don't have to fucking put your paws on that. Yellow.
I'm here.
Oh, I don't know. Better than the markets? I don't know.
Is there anybody that has any questions?
Questions or comments?
Hey Scotty, Belgian Crider here. I got a question for you.
Yes, Bill.
On the lignojule, what is the research done when you put it on the seed?
Okay, on pulse crops, I've had significant yield opportunities on peas, lentils, chickpeas would be my primary one, where the yield is very significant. I've had peas in the field as well as in the research that will generate 12 to 14% on a regular day in, day out basis. On soybeans, however, unfortunately, they're a really hard crop to push, and I see generally a 5% increase from the application on seed on soybeans, but then I can stack it. I can generally add another 5% on top of that with one foliar application, usually with a late herbicide and or at R1, if anybody's going out at that stage. Thanks for bringing that up, Belgian. It is very significant on pulse crops. On phaseolus beans, which are your edibles, it's like fava beans, kidney beans, it's not near significant. I don't generally promote it much on those particular crops. On a cereal crop, if I'm under irrigation, I'll attempt it and I'll use it marginally because I'll grow a beast. And if I grow a beast in a cereal crop in Western Canada when we turn quiet and dry in July, August, if I have too much canopy in a cereal crop, I can actually lose yield when it comes to setting seed. So generally speaking, it's not promoted on cereal crops. Pulse crops primarily. Thanks, Belgian.
Does anybody else have any questions for Scotty?
Scotty, Blake
Yeah.
here. Any results on the ACF product foliar applied dealing with those salinity spots, or just an in-furrow thing?
The foliar applications have been done through irrigation, and yes, very significant. We did some potato work out west where it was put through a pivot. It was actually broadcast prior to emergence, and then it was run through a pivot, and very significant. 2 applications. The first one was 3 gallons. The second one was 2, or maybe even more from what I'm learning. But this whole salinity thing, it's going to be one of those walk before I run, because right now I'm of the mind to say my primary application right now is in furrow. And that's why, as you may know, Blake, I've been focusing on a lot of alpine users because they have kits on the drill. I can get it away from the sunshine. I can protect it. I need to get it onto the soil surface. So I guess in the eastern part of Manitoba, I might be able to get some applications and trial work to get it on soil. But I know that if I put it on, my primary application is in furrow to get it away from the sunshine. If I put it on a whole bunch of crop residue, I'm afraid of loss due to the ultraviolet sun rays killing it. So until I can go down the road and get more definitive things going, I know there are a number of chickpea and pea growers further west that actually apply it in season over top of the crop to change that soil environment. And it makes significant differences throughout the season for plant health and the disease suppression. But it's going to take a lot more work to prove those points that we can maintain and subdue that disease from moving from the soil to the air. We're going to be able, I'll retire on the research of this product. I know it. It's going to be in depth.
Okay, thanks, Scotty.
Is there any other questions? Well, I think we'll take it from there and wrap it up. What do you think, Scott?
Yep, thanks very much for everyone's time. I hope this was an informative and it breeds a lot of questions and interest, so that as we move down the road of creating more and more opportunities with these materials, we learn more and more about them as a team. Thank you.
Thank you, Scott, and thank you everyone for attending, and hopefully you'll stick around for our next webinar here in 15 minutes. Thank you.